Welcome back. It's Intro to Physical Anthropology. I'm your instructor, David Leitner, and today we are going to talk about human variation and natural selection. What are some of the ways evolution has shaped our genetic and phenotypic diversity in this world? So, without further ado, let's get started. Okay, so the first concept we need to sort of come to terms with is this notion of polymorphisms. Poly means many, morph means shape or form, so it's many forms. And all it means is, you have a polymorphism when you have two alleles or more in the gene pool. So, any genotype or phenotype that's present at above 1%. It's sort of an arbitrary number, but not that arbitrary. But anything above 1% in the population is considered a polymorphism. Why 1%? Well, at about 1%, it's less likely to just be a sort of one generation sort of fluke. It's less likely that it's going to sort of go off in genetic drift. Usually, it depends on the population size. But either way, a polymorphism needs to be at above 1% in the population. Now, a great example of polymorphisms is the ABO blood system. We're coming back to it. We talked about this in another context before, but ABO blood types can be mapped clinally. That means you can map them on the globe or on a map based on their changing frequencies. So as you can see here, we've got gene frequencies for the hemoglobin A phenotype or genotype that are going in roughly 5% increments here, and you can sort of see that it's mapped across the globe. Now, that's true for the others as well. Interesting thing about the ABO system is that depending on where you are in the world, the frequency of that genotype will be different. So if we're looking at it's most common in Europe, Australia, parts of North America, Greenland, fairly common in Africa and Asia, and fairly absent in Central and South America. If you look at the type O variant, you see that it is mostly prevalent in North and South America. And for the type B, it's mostly prevalent in Asia and parts of Africa, creeping into Europe, but mostly those areas. So, O is by far the most common worldwide. It might not look it by comparing these maps, but remember the percentages are very different. So gene frequencies, the lowest gene frequencies mapped on gene O are 50 to 60%, whereas the highest frequencies for A are above 35 and 25 to 30 for B. So O is worldwide the most common, but when we actually look at the map in Asia, it's not as nearly as common as it is in South America. Then you have A, and then you have B. So there are some theories about why O might be so prevalent in the Americas, and they could be for a couple of number of reasons that aren't necessarily mutually exclusive. The first is founder effect. There just may have been a disproportionate number of type O individuals in the original populations that first migrated either by coast or across the land bridge across the Bering Sea. That's a very, very good possibility. The other is that selection might actually play a role. There's been some correlation between type A and type B blood with bacterial resistance, and some correlation with type O for viral resistance. So perhaps early on, or when they arrived in sort of different places in the Americas, the early Native Americans encountered more viruses than they did bacteria, and so the viral resistance became more of an advantage. It could be both. It could be both of these explanations. But the point is, these maps, these clinal maps, are shaped by evolutionary forces, right? Okay, so we can have different kinds of polymorphisms. The first one is a balanced polymorphism. This doesn't mean that they're present in equal amounts. It means that the polymorphism is apparently stable in the population. It's neither sort of rising in frequency or decreasing in frequency as a result of natural selection. Something is keeping the polymorphisms at what we call fixation. It's keeping them from reaching fixation. Fixation is when one allele completely dominates the other one to the point where the other one disappears. An example of balanced polymorphism is sickle cell. The frequency of sickle cell in malarial regions is balanced because of heterozygous advantage. This is the fact that the heterozygotes, those who are carriers for the sickle cell trait but don't have the sickle cell trait, have an reproductive advantage over individuals who don't carry the sickle cell trait, even though offspring they have with the sickle cell with sickle cell anemia will not probably survive in the past. It's more likely now. The reproductive advantage is still there because their children will be, their remaining children will be resistant to the malaria parasite. So there is a balance there. If sickle cell overtook the normal hemoglobin, then that population would die out. If the normal hemoglobin overtook the sickle cell, then there would be huge amounts of death from malaria in the population. But this heterozygous advantage, or hybrid vigor, as it's sometimes called, helps to keep the frequencies of the polymorphisms balanced. Now, some polymorphisms are only advantageous at above or below a certain frequency. We call these frequency-dependent polymorphisms. So fitness is determined by how much of the other polymorphism there is, or how much of the polymorphism in question there is. There's positive and there's negative frequency dependence. So, positive frequency-dependent selection is when the phenotype is only advantageous when it is maintained above a certain frequency. An example of this is the mimicry of king snakes for the coral snake, the venomous coral snake's coloration. Coral snakes are venomous, and they advertise this with this bright ring patterns of red, yellow, and black. King snakes mimic this. They have a coloration very similar to it to trick predators into thinking they are venomous snakes, so that predators will stay away. However, if there aren't enough coral snakes in the region, what we find is king snake coloration varies considerably among individuals there because it's no longer an advantage. The predators need to encounter coral snakes often enough to learn the lesson not to mess with them in order for the king snake's mimicry to benefit the king snake. So the fitness of the king snake's coloration depends on the frequency of the coral snakes in the region. Now, negative frequency dependent selection is when the phenotype is only advantageous when it's below a certain frequency. great example of this is the relationship between the song thrush and the Cepia nemoralis snails. I think I got that right. Cepia nemoralis has various shell patterns, as you can see here in the bottom picture. They're really beautiful. But the song thrushes in their territory will tend to have a search image, it seems, where they have an image of the shell pattern, usually of the most common type of Cepia in that region. And so individuals with that snail pattern are the ones that the song thrush is going for. So if you have a variant that doesn't look like that, you are going to have an advantage over the other snails, because the song thrush won't be looking for you. Now, that advantage disappears if your version of the shell pattern becomes too common in the population, because then the search image changes, and now you're the target. So there's an example of needing to keep the frequency of the polymorphism below a certain trait. So it's a negative frequency-dependent polymorphism. Now, not all polymorphisms are the result of, well, they're all the result of natural or sexual selection and the other forces of evolution, but some are the result of evolution working through culture. That is, culture becoming a selective force, or provide in such a way that it changes traits in human beings. We have a number of these that we can point to, but the most common one is the trait of lactase persistence. You've heard of lactose intolerance. The fact of the matter is, lactose intolerance is the norm for most mammals. Adult mammals are not supposed to be able to digest milk. Lactase persistence is the mutation, and it is something that has appeared multiple times in groups of humans. It has only become prevalent in some groups because of changes in culture. And I'll expand on that in a minute. First of all, lactose is the sugar in milk. Okay? Lactase is the enzyme that infant mammals produce that allows them to break down lactose into sugars they can digest. As you are weaned, you lose this ability to produce lactose, so you produce less and less and less of it. And so adults should not be able to drink milk without having, if that lactose is in your gut, but there's no lactase to break it down, then bacteria in your gut break it down instead. And when the bacteria break it down, they produce gas, which can create bloating and ultimately diarrhea. Very serious cases of diarrhea. I will admit, I am one of the lucky norms here who does not have lactase persistence. So believe me when I tell you it's not fun. Now, interestingly, it occurs most frequently. This polymorphism occurs most frequently in indigenous populations that have historically herded. So groups of people who have lived with animals, domesticated animals, tend to have this trait associated with them. So Northern Europe has a very strong correlation, but there are also strong correlations in East and West Africa, the Middle East, and India and Pakistan, right? These are all cultures that have long histories of herding either cattle or goats or other mammals, and of drinking their milk. Now, a little diarrhea under normal circumstances isn't a big deal. It's temporary, and you'll be okay the next day. But during times when food is scarce, diarrhea can be fatal. Absolutely fatal. You're already working on reduced calories. Now you've not only taken in something that you can't digest, it's going to force all of the digestible matter in your gut out. So you won't get as much from that either. So it is a huge reduction in your caloric intake. It has a huge impact, not to mention dehydration and to be honest, you aren't as mobile. You can't get around. So during these famine times, we think is when the selective pressures kick in. So people with the lactase persistence gene have another source of calories they can call upon that people without it cannot. So they are more likely to survive, even if it's only by like 1% more likely. That has a huge effect over time, over several generations. And you will see this trait explode in the population. Pardon the unintentional pun there. But this is why it's a great example of gene culture coevolution. The selective pressure only kicks in if you are in a herding community. We actually know that all the places it's cropped up, it's been a different mutation in the same gene, but in a different place. So we think this gene has constantly been kicking up these lactase persistence mutations over time, but it's not until you have a culture that drinks the milk of other animals that it becomes an advantage. So gene culture evolution, coevolution. Okay, now we can talk about traits in terms of adaptation, which is the result of natural selection or other evolutionary forces, like we have been. But we can also talk about it in terms of adaptability and acclimatization. Our bodies are flexible, especially human bodies. We're very good at adapting to multiple environments. So let's talk about the differences between these very closely related terms. Adaptation are changes to the genotype. The other two do not change your genotype. Adaptation changes are what we've been talking about with selection, right? And they are the result of anything that confers an advantage or disadvantage in differential reproductive fitness. We've talked about how skin tone has evolved, and that is one example of it. Another is certain populations' reactions to living at high altitude. In two places in the world, there are groups who live above 10,000 feet, far above 10,000 feet in many cases, who have adapted to a lifestyle that has a much lower available oxygen content in the air. So they have adapted to lower oxygen levels. And the adaptations are completely different. But it is through selection that this occurred. Okay, the first is that the baseline hemoglobin concentrations, that is the oxygen carrying cells in the proteins in the red blood cells, are increased in Andean populations. So the Andes are one place people live above 10,000 feet. The Himalayas are the other. In Andean populations, we see an adaptation of having a higher baseline concentration of red blood cells. In both, we see a less pronounced hypoxic ventilatory response. This is the thing that makes you start breathing a lot and can lead to altitude sickness if you go up too quickly. And their reactions to this response are better regulated. This is even more pronounced in Andean populations than it is in Himalayan ones. Finally, Himalayan, Nepalese and Tibetan people have some resistance to Mung's disease, which is also known as chronic mountain sickness. If you go up to high altitudes and aren't adapted to it and live at extremely high altitudes for long periods of time, you will develop some very negative health effects as a result. Tibetans seem to have some resistance to this. Okay, so those are examples of adaptations, but we also have acclimatization responses. These are short-term changes to individual phenotypes in response to environmental conditions. They are short-term, and they only occur to the individual. They aren't passed on to the next generation, except as part of our normal human biological inheritance. And once the stimulus changes, the trait will turn back. The phenotype will turn back. Hypoxic ventilatory response, although it is better in Andean and Himalayan people who live in the Himalayas, it is something all of us have. So the HVR starts with increased heart and breathing rates. And that's because with less oxygen saturation, in order to get enough oxygen into your bloodstream, you have to get more air through your lungs to get as much oxygen. So your heart pumps the blood faster and your breathing goes up and becomes deeper so that you can get more oxygen in. But it has some negative side effects like headaches and tunnel vision, etc. if it happens too quickly. Longer term responses include if you're up there for, a week, you will start to have increased hemoglobin concentration. So your red blood cell count will go up as a result. And you will notice that the effects of HVR will start to subside as a result. And then once you're sort of acclimated to that altitude, you'll be able to sort of go about a little more freely. So our bodies have this ability to temporarily change our physiology so that we can to accommodate these environments we find ourselves in. Altitude, of course, is one form of acclimatization. Heat, vasodilation, the fact that the blood cells in the outer parts of your body relax and open up when you are hot so that you can release more of your core body heat is part of a heat response that is an acclimatization. Cold, when you shiver, when your blood vessels constrict, when you get goose pimples. These are all examples of acclimatization. Although the goose pimples don't help us as much as they did when we were fully covered in hair. But caloric intake, when calories are too available, or certain kinds of foods are too available, or when they're not available enough, body weight changes. Your body accommodates those changes. Ultraviolet radiation, tanning is an acclimatization response. It's not a permanent change to your skin tone, but it affords some temporary protection. Finally, we've got adaptability. Adaptability is either a short or long-term change in phenotype as a result of individual physiological plasticity. Actually, oftentimes these are very long-term and possibly permanent changes, because they are changes to the development of the organism. The environment influences the organism during development, and those changes last. Inadequate nutrition during development. You can get diseases like rickets, which will deform the bones. Same with marasmus. Excessive weight gain as a child can have impacts on the health of your vascular system later on. Intentional body deformation, so corseting, extreme corseting, the kind of corseting women normally did in Victorian times wouldn't actually, these horror stories we have of them dying because of their organs had been constricted too much, etc., etc. Those are mostly apocryphal and probably not true. But we do have examples of women who did extreme corseting from time on a regular basis, who did deform the shape of their rib cage as a result. infant cranial deformation or manipulation. This is common in several cultures. It was very common in some Mesoamerican cultures where an elongated skull was seen as a sign of royalty. And so the highest status people would do this to their children so that they are, like themselves, recognized as high status. And finally, if you grow up at high altitudes, you may develop some permanent increased lung capacity. Last thing I want to talk about today is what some of these changes, these physiological changes that occur, mean for evolution. It means that there are adaptations we can see in skeletons that are going to be important later on in this course that tell us whether this is a cold adapted or warm adapted species. The first is Bergmann's rule. Actually, I should say population, because we see these in different populations. Bergmann's rule says that in cold environments, you will have a larger body volume, but lower surface area. This is because, so that means you're going to be sort of stockier, probably shorter, not necessarily, but you're going to be stockier. This is because the advantage is if you have low surface area, you have fewer places to lose heat. So that's a cold adaptation for conserving heat. Allen's rule, on the other hand, says in warm environments, you will see people like the Maasai down below. Those are Sami people from Norway. You probably didn't know there were indigenous people in Norway. These are the Sami. And it's obviously an old picture from 1900, but this is their traditional way of life. You can see, unlike the Maasai, they have relatively short legs compared to their body size. The Maasai have very long legs to their body size, to their torsos. Warm environments encourage this, in part because a tall, slender profile probably also gets you into cooling breezes more. It actually means there's less surface area for the sun to beat down on if you're in an equatorial environment, and creates more surface area for cooling in general. In cold environments, again, to conserve heat, short limb to body ratio is it kind of goes hand in hand with Bergmann's rule. It may also be due to the fact that there is some influence of temperature on cartilage growth. So it may be not just an adaptation, but may also be adaptability, which is what we were just talking about. So an effect on development. Okay, hopefully that has set us up so that we can start having good discussions on how these factors, these forces of evolution and genetics, will come into play when we start studying human evolution. We need to know these things so that we can infer from the fossil evidence what people were like, what their environment was like, and how they might have lived. That's the reason I've spent this much time just teaching you the basics of evolution here. Because we can't understand the rest if we don't understand this. That said, thank you very much again for your time, and I hope you are staying well and taking care of yourself. And I'll see you again soon. Bye-bye.